Good afternoon, everyone. Thank you for joining us at this year's Canaccord Genuity Musculoskeletal Conference. My name is Michaela Smith, and I'm on the MedTech team here at Canaccord. I'm joined today by DiscGenics, a privately held biopharmaceutical company focused on developing regenerative cell-based therapies that alleviate pain and restore function in degenerative disc disease patients. With me today is Flagg Flanagan, CEO. Thanks for joining me today, Flagg.
Yeah, thanks.
Before we begin, I want to remind everyone of any relevant disclosures which can be found on our conference and our firm website. With that, I'll hand it over.
Great. We're good to go. Thanks, Michaela. Really appreciate it. Appreciate Canaccord inviting me. Very happy to be here to give you an update. I'm going to deal in some generalities because we have some confidentiality agreements ongoing with some of our major clinical sites for our Phase 3 and other things, but I'll try to be as specific as possible and then answer questions afterwards. We are currently a late-stage clinical biopharmaceutical company focused on developing regenerative cell-based biologics. Our lead product is injectable DiscGenics cell therapy for lumbar degenerative disc disease. Target patient population is about 2.5 million patients annually in the U.S. alone. I know you find that to be a big number, but to give you kind of a corner market comparison, there are 2.1 million patients diagnosed with cancer in this country. We've shown that a single injection of injectable DiscGenics cell therapy has demonstrated regenerative capacity and the potential to treat underlying pathophysiology of degenerative disc disease, both in improvement in disc architecture and reduction in inflammation and pain in an IND-allowed Phase 1/2 clinical study, and that is an IND, investigational new drug. We've been granted special regulatory designations, regenerative medicine advanced therapy, which allows for potential accelerated approval and priority review based on our Phase 1/2 data, and a fast track designation in 2019, which enables a rolling biologic license application, which is very important because we received that application before we ever injected a human patient, because we used all human cells in our animals to show the immunoprivilege of our allogeneic product and the safety of the product. We currently have a clinical dose manufacturing in process in an in-house scalable built-for-purpose allogeneic cell facility, CGMP, in Salt Lake City. Love to have you come by and visit as you pass through Salt Lake. Delta's a good hub there. 56 issued patents through 2024. And a couple going longer, covering 4 primary patient families around IDCT and the DiscGenics cell CGMP manufacturing process, with 34 more pending and many, many more trade secret protections around our manufacturing. We have de-risked this clinical pathway with an FDA-approved through the CBER branch Center for Biologic accelerated Phase 3 program, utilizing a parallel pivotal and confirmatory study. A little bit more on confirmatory, y'all may have heard some stuff about that, to target pain and disability. And we're doing this as a treatment for degenerative disc disease. We had the first patient treated December 23rd of 2025. We have an Asia initiative going on in Japan. We've been working on that for about 12 years with a safety study that were results that were consistent with the U.S. study. So we really are pretty satisfied about where we're going with these 2 studies and where it can lead. We are currently working with the Minister of Health, Labor, and Welfare in Japan around a Sakigake designation. That is really for more life and death designation, but they're willing and they asked us to apply to be the first chronic possibility here. So we're pretty excited about that and working with the PMDA on a possible conditional approval. So our history, it's a fairly long one, but you can see how we spent a lot of time doing a lot of preclinical work, which resulted in an IND and raising of money along the way, and finished that IND Phase 1/2 study. When then had a $50 million Series C round where we built out our manufacturing facility, did a study in Japan with that last patient in April 2021. So we did determine that the high-dose patients did meet the primary endpoints, got the RMAT designation, research and collaboration agreement with UPenn I'll talk about on a total biologic disc, and really continuing great results out of our high-dose IDCT, which was published in the International Journal of Spine Surgery, and we're currently working on our Series D round of funding and a pivotal arm and the confirmatory arm as we start our Phase 3 program. As you know, it's a huge patient population, pervasive, 16 million patients in the U.S. every year, leading cause of disability, easily $100 billion more spent in the U.S. alone, primary reason for non-cancer opioid prescriptions. As you can see, L4-5 is a normal disc with the degeneration at this single level of L5-S1. We think that for us is a very treatable disc. Current treatment options for symptomatic lumbar DDD, fusion for predominant low mechanical back pain has suboptimal clinical outcomes, while artificial lumbar disc replacement has been good in cervical, not nearly as good in lumbar. Current treatments for symptomatic DDD, only short-term limited benefits and do not result in repair or regeneration. And ideally, it would be best if we could restore function and disc structure biologically. And as you can see, if you look at the modified Furman grading scale, we have shown success in the range of grade 1 to 8. The total, we've shown very good success, grade 3, 4, and even out to grade 7. So we're pretty excited about where we are with our product and where we're going. And we showed that an injection of injectable DiscGenics cell therapy, generic name Rebonuputemcel, into a painful degenerated disc is really designed to treat the progression and halt it of DDD, but also regenerate the disc from the inside out. And we use this live progenitor cell population, very unique, derived from donated human adult intervertebral disc tissue. We are not an MSC. We are not derived from adipose fat or bone marrow. We are derived from disc tissue where we do a cell selection. We do a selection and expansion. We then freeze that donor lot down, and we accumulate these donor lots where we do testing, which we show purity and potency and comparability across donor lots to the highest CMC standards, according to the FDA drug and biologic standards. And we then take that accumulated those lots, we take a lot out, we put it in a 250-liter bioreactor at which we derive somewhere between 2,500 and 4,000 doses. So it's truly a pharma margin type product. This active ingredient of live progenitor cells derived, it's cultured as we use growth factors to drive it back to a progenitor state to maximize potency, and it's frozen to ensure viability and through cold chain logistics that we showed in 14 sites in our US study and 7 sites in Japan. It's injected into a disc. It's a very simple procedure in an outpatient setting in a procedure room utilizing a C-arm for guidance. It requires no donor matching or immunosuppressants, which again was why we got our fast track designation, because in using all human cells in our animal models with no safety issues or adverse events. So I want to do this slide. It's a little busy, but I want to take you through it because it really, we spent a lot of time for the disease state to inform MOA and clinical endpoints. And we really looked very closely to understand the pathophysiology of degenerative disc disease, which is inflammation causing pain, and this degenerative change, which is basically an imbalance between the anabolic and catabolic environment of the extracellular matrix, leading to loss of disc hydration and the disruption of disc architecture. So we spent 7 years doing 14 different animal studies to really understand and really vet out the purity, potency, and comparability across donor lots to submit for our first IND for investigational new drug to start the phase 1/2. And you could see that we showed very good lack of immune response despite the use of animals, of the use of human cells in the animal. And we saw that the extracellular matrix, which was secreted from these cells, rebuilt the disc architecture and improved it dramatically. And right at the end, just before our IND, the FDA was pretty adamant about us doing a live tumor genicity model because there had been a lot of issues with tumor genicity with cells in the past. We did this model in an athymic muscle pouch study, which I'm sure many of you are very well aware of, and not only did it not grow tumors, it spontaneously grew disc tissue. So we knew we were onto something in a big way through our animal studies, and we developed 2 MOAs around our product. First, anti-inflammatory and the secretion of anti-inflammatory cytokines, and then a regenerative capacity through this secretion of extracellular matrices resolving primarily around aggrecan and collagen production. And then we had the very positive patient-reported outcomes through our high dose in our phase 1/2 study, where we showed pain reduction, functional improvement, and improvement in quality of life in the US and Japan. And then we saw this never been seen before disc volume improvement in both the US and Japan that really truly is indicative of true regenerative capacity. And first and foremost, just as we had seen in the animals with human cells in humans, we saw no treatment emergent serious adverse events associated with the drug product or the injection. So today, we have been signed off by the FDA to start a phase 3, which will be an accelerated phase 3 of pivotal confirmatory. It will be as a treatment for degenerative disc disease with pain reduction and functional improvement and disability, followed by quality of life, disc volume improvement, and reduction in pain medications. And of course, it'll follow with safety as well. So we're very excited. Just to give you a recap on our phase 1/2, we had 4 arms in this study, a high dose of 9 million cells, low dose 3 million. We used a vehicle control, which was our sodium hyaluronate, and a placebo control, which is normal saline. And as you can see, the high dose immediately within 12 weeks shows a dramatic reduction in pain, which sustains itself durable through 2 years. And you can see the normal saline functions pretty much as normal saline, which is a known therapeutic that in the first short term of 26 weeks, it does work pretty well, and then it fell away. There's a wasteland of disc injection products that have never been able to size up to normal saline injection. And I think we did a pretty good job of differentiating ourselves here with the high dose. You can see that the low dose shows some effectiveness along the way and does very well. And then we were a little concerned with the vehicle control, the sodium hyaluronate, but what we saw is while opioid use decreased among the high dose IDCT group, they increased in the vehicle control group compared to baseline. So they were taking more drugs to keep the pain away. And then when we looked at ODI, you can see the high dose definitely runs away from the other 3 arms in a good way. We used MCID, minimum clinically important difference, to do a within group comparison because we were not statistically powered to do between group comparison. But you can see with the high dose, we showed 1.97 MCID increase over the ODI factor, which was very good. And as good as that was in quality of life, we were almost at 3 times MCID with the high dose and statistically significant at all of our time points. And then we saw this whole increase in disc volume of the high dose as it did very well out to 250 cubic millimeters at 52 weeks, and then up to 400 cubic millimeters at year 2. And it's very interesting if you look at the other 3 arms, you can see that the low dose continues to maintain disc volume. So we saw some effectiveness, but we saw a dramatic difference between the 2 arms of the high dose versus the low dose, which allowed us to use the high dose in this next accelerated phase 3 study. And then you see also that the placebo and the vehicle control drop dramatically below baseline. Case study real quick of a high dose patient, lady, single level in a car accident, L5S1, had a small partial posterior annular tear with a loss of disc height. You can see 6 months after MII and injection, disc height is improved along with volume, clearly increased. And you can see Also, where the disc volume increases, there is some annular repair. And what's most important is, along with this increase in disc volume and the healing of the annular repair, look at what happens to the patient-reported outcomes. A VAS goes from 47 to 4. She was taking a leave, kind of like M&Ms, all day long, and her ODI went from 44 to 16, so it was in the third quintile back to the first quintile, which is no disability. Her EQ5D, her quality of life was pretty good, but it also went up to 90. This patient is now out to 4.5 years. She has had an additional 50 cubic millimeters of disc volume improvement that happened really at about year 3, and it has settled out. And we always thought that there was a certain space inhibition with these cells, that once they hit a certain pressure point or a maximum point, they would subside and level out, which has been the case with this patient. But she has great durability, and her patient-reported outcomes are the same. We had a peer-reviewed published study through the International Journal of Spine Surgery. It was their best paper in 2024. It was the most downloaded paper in the history of the International Journal of Spine Surgery, and it really brought a lot of people out after reading this paper. Major spine centers that are very interested in this because they really don't have anything they can really do to a mild to moderate patient. And they think this could possibly be a treatment for it. So we took this data back to the FDA. They approved an accelerated phase 3 study to run the pivotal confirmatory in parallel, 324 subjects, 216 IDCT, and 108 sham. So it's a 2-to-1. Endpoint structured to show full treatment effect of IDCT for DDD, 52-week primary endpoint, and a 104-week closeout, exactly the same as our phase 1/2. So we're very excited about where we are there. We are contracting with a really very interesting stratification of sites for our phase 3, and we think it's going to be very exemplary of sort of the who's who in spine from an institutional and integrated spine practice standpoint. We are utilizing some pain management and some pain and ambulatory surgery combined centers, and several university integrated spine. So you can see it's a pretty good stratification of these institutions, and we've done them to a regional standpoint because these will be the first centers that will convert when we commercialize. So we're pretty excited about those. I can't talk about other than Dr. McHale being with Cleveland Clinic, but it will be the who's who in the spine world that will be our clinical sites going forward in the phase 3. We have a vertically integrated internal CGMP manufacturing facility in Salt Lake City. We take about a quarter of this overall building, and very excited about what we've done here for enabling full-scale lot production capabilities, support commercialization, and we really control our destiny by vertically integrating. And it reduces cost and improves margin. It protects and expands proprietary intellectual property, especially as we optimize the commercialization and the manufacturing of this cell. We're starting to learn a lot of cool things that think will go far above that 2,500 to 4,000 doses per donor lot. Robust quality management system, and we believe it accelerates the overall value proposition 30 to 50% easily. As you know, in drug, the manufacturers take about 30 to 40%. So IDCT, we believe, unlocks a larger spine total addressable market. As you can see, $16.6 billion spinal fusion by 2032. Our friends at Bruder Consulting came out at $100 billion estimated total addressable market by 2035. A lot of people gasp at that. There's some people in this audience I see who are very well versed in this space nodding their heads. And I'll tell you, even if they're 50% wrong, I like my chances. So we think that we are poised to help literally millions of people around the world with something that is truly regenerative and will be an effective treatment for DDD in the mild to moderate space. We do have this IDCT, these discogenic cells, as a platform technology around lumbar, and then we'll go to 2-level cervical adjacent segment, post-discectomy, possibly annular repair. We go very well with Barricade. Saw his presentation earlier. We are looking at another product for articular cartilage degeneration. We have done some work on a total biologic disc that I'll talk about, and facet joint regeneration. So this is the EDAP's biologic disc replacement, and this would be a total biologic disc for cervical. And the key findings of this exercise we did through the University of Penn and the VA, and we showed that manufactured with discogenic NP and AF cells, that's nucleus pulsus and annulus flows, were mechanically and biochemically distinct from MSC-seeded EDAPs. It's suggesting superior functionality. So it really is interesting, and through these animal studies, we did show that the living human-sized disc withstood the mechanical testing and produced region-specific extracellular matrix consistent with the natural dysfunction. Still a lot of work to do, still early. A lot of people told me in 2008 and 9 that I said I was going to work to build a biologic disc. They thought I was a little crazy doing that, but I think this total disc is a reality that will happen. And the beauty of this is a picture is worth 100,000 words. You can see the proliferation using goat MSCs and human MSCs. And the human MSCs, you don't see much proliferation, but look what happens with the discogenic cells relative to the proliferation over both the goat MSCs, and from, and we use the discogenic cells 2 ways, from attachment and from suspension. So you can see that wonderful proliferation. And we have another cell platform that does make more collagen, which we're excited about to use around other musculoskeletal body parts of the body. And it is a true allogeneic. It is totally immunoprivileged for those joints. So we're working on that, and there's some testing underway for new delivery platforms to be able to nest those cells in cartilage defects to enable new musculoskeletal information. And, musculoskeletal indications, sorry. Next steps, FDA-approved Phase 3, the execution on that, selection and onboarding underway, clinical CGMP manufacturing in our own built-for-purpose manufacturing facility for Japan and other parts of the world. First patient treated, Q4 2025. Convertible note, Series D crossover in process. And very quickly, because I want to get some questions in, key differentiators, we're standalone, novel, proprietary, unique cell therapy that does regenerate the disc. We do have CBER, biologic branch approval on CMC clinical protocols for this Phase 3 enabled study and accelerated pathway. We de-risk it, first patient treated, in-house manufacturing serves as a platform, follow-on allogeneic cell platform for other musculoskeletal indications, and the total disc. We had the first patient in, and we were honored to be noticed by NASDAQ on the Jumbotron in Times Square. So we're off to a good start. I got a minute and a half to either take questions or ask y'all a couple. Thank you. Questions? Yes.
Could you speak to the ICCT program coming behind IDCT?
I'm sorry.
The cartilage.
Yeah, so we have identified another allogeneic cell that produces more cartilage, and we believe that that can be applicable for cartilage defects in various other joints. So we're hopeful for that. It's still early. We're looking for carriers that will hold those cells in place. So it's still a ways away, but it's an opportunity. Working up and down the disc, it gets exorbitant. And the brooder piece is really around lumbar for that estimation of total addressable market. So you can imagine what this could be. I mean, it gets to be kind of crazy to get your arms around what the total addressable market could be. Anything else? I got a question. How many biologic products are approved for spine through the CBER branch? Anybody know? Anybody care to guess? Do I hear 10? God, I got some good people. 0. 0. They're all IDE or other things for fusion, et cetera. How many biologics are approved for musculoskeletal? One, Vericel, the Macy. It's an autologous where you have to go in and do a arthroscopy, nip off some cartilage cells. They send it back to the factory where they expand it, put it on a porcine graft, and then put it in a cartilage defect. And right now it's at open surgery. They soon to be able, hopefully, to do it arthroscopically, and they get somewhere between $65,000 and $80,000 for that total procedure. So I think it bodes well for us to have something that's allogeneic, that's off the shelf, that we can thaw, draw, and inject. It will come from our freezer to the clinic freezer and be available immediately for use. So we're very excited about the opportunity. We're starting out at 1 SKU. We don't think it's very difficult to manage a Mayo stand in a procedure room. So it really will be more of a delta force of pharma sales executives that really work on accretion of patients to those integrated spine practices and high volume pain practices as we go forward. I've used up all my time, and I'm deeply appreciative for your attention. Thank you.